He received one of our very first research grants years ago, and after finishing that work, he applied to the NIH and received a huge grant from them to follow up, and he's still continuing his genetic research, and he is the one who's discovered a lot of information about PSC genes, so we are forever indebted to him, so thank you. So, also, we have, would like to give the PSC Partners Partner of the Year Award to Dr. Lazaridis, Eaton, and Andrea Gossard. I think you're the only one here today. Okay.
Thank you so much, Ricky.
Thank you.
Thank you. Good morning. On behalf of our Division of Gastroenterology and Hepatology, our research groups, and the Mayo Clinic, I would like to welcome you to Rochester and to Mayo Clinic for your annual PSC Partners Conference. It's our distinct honor and pleasure to have you here today. We'd like to let you know that you are an inspiration for us for what you do, and your work motivates us to do more work to better understand and treat the disease called primary sclerosing cholangitis. I would like to share with you a story that started about 15 years ago about my own work in PSC. Few of you, 2 days ago, you visited the Mayo Clinic Biobank, which is a magnificent facility by any means, one of a kind in the US, where we store several millions of biospecimens as we speak. But 15 years ago, that was not the case. When I started doing the work in collecting biospecimens, we used to have a very small lab in the very basement of the Department of Laboratory Medicine, where they were collecting biospecimens for investigators like me at no cost, and they were doing the best they could. Actually, they didn't have enough space to store the specimens for a long time after a month, and so after a few months, when I started to collect biospecimens, they called me back and said, Dr. Lazaridis, you have a box of about 100 specimens. It's time to come and pick it up. So I did there. I picked my specimens, which was your specimens, and I felt very emotional because for the first time I realized that as I'm going to my laboratory to do work on those 100 specimens, I have now 100 people who trusted us with their biospecimens, and I know that they come from different backgrounds from all over the US, and they look for answers. And I knew and I know that on those specimens, I had some answers. So this emotional feeling about what you can trust us to do keeps motivate me and all of us who do work in the space of PSC. So thank you for doing that. Thank you for your support. I'm grateful also to all the faculty who are from Mayo Clinic or from the outside to come and talk to you about their expertise for this disease. I'm grateful to Dr. John Eaton, to Andrea Gossard, who were in the committee to organize this conference. I'd like to thank Ricky and Megan. They have done tremendous work over the last year to make this an outstanding meeting. And finally, I would like to thank my secretary, Jen Root, who have worked tirelessly for the last 6 months or so to make sure that we have everything in place. Finally, I'd like to thank Irish Link and Jackie, my study coordinators, who make possible for me to still be able to collect biospecimens, which is critical for our research and for our work. Thank you for your time. I hope you enjoy the conference, and I hope this is a productive time for you, for us to answer questions and to make this an experience so you can go back and feel like we do all the right things to do to make the therapy or the understanding of PSC something that we're looking forward in the future. Thank you so much.
All right, good morning, everyone. My name is Robert Hebert. I'm a physician scientist and a transplant hepatologist at Mayo Clinic. And my work focuses on epigenetics and regenerative medicine as it relates to PSC. And I'll be moderating this first general session, which is on genes, environment, and animal models in PSC. So we have three excellent distinguished speakers for you this morning with short talks, and we'll hold the questions until the end. We'll invite all three speakers up for simultaneous questions at the end. So our first speaker is Dr. Lazaridis, who you know, professor of medicine at Mayo Clinic, expert in large-scale gene sequencing and genetics. And today he's going to tell you about a fascinating area called Exposomics and how the environment may interact to cause disease. As soon as we get his slides up. Wrong talk. Could be. Nope, same one. There it is. All right, Kostas, the floor is yours.
Robert, thank you for the kind introduction. Now, when you ask a Greek to give a talk, usually they will talk about history and mythology and something about it. So you stuck with me. You're not going to, and you're going to hear a number of things about genes and environment, but I will be philosophical and historic at times of my talk. That's how I operate. And actually, this is the first title of my talk. This is what we knew about PSC about 30 years ago. This is what we knew about 20 years ago. This is what we know today. And this is where we want to go. This is what we would like to know 5, 10 years from now. We like to be able to have a better picture of what causes this disease and what we can do to treat this disease. Now, as I said, I like history. So the first ever report coming from the Mayo Clinic about primary sclerosing cholangitis was in the 60s, actually. It was by Dr. Bartholomew, one of our staff in gastroenterology. And then, of course, this is a landmark paper by Drs. Wisner and LaRusso, where for the first time, they reported the first 50 cases seen at the Mayo Clinic back in the 1980s. That was a time where a new era, I would say, for PSC started, because we were more organized about following this disease and developing a lot of work into that space. I joined the Mayo Clinic as a resident back in 1993, and I had the great pleasure and opportunity to work with Dr. Gores, who was my advisor since then. And he has been giving me advice, which was always on target. And he was the one who convinced me to work on primary sclerosing cholangitis at the very beginning. He was also with Dr. LaRusso, who told me, you have two choices, Kostas, as you're done with your fellowship as far as what you do next in your life and develop a career. And they gave me two options. One was to study the genetics of a disease, and they were thinking about primary sclerosing cholangitis. The other was to study hepatitis C and the related virology. I chose the first in a heartbeat, and I haven't regretted this decision 20 years later. Time goes fast. So back then, it was 1999 when I met Nickie and Don for the first time. There was an NIH symposium at Bethesda. Nickie remains the same, energetic, devoted to PSC. Don remains the same. He improved his jokes over the years. They are more politically correct as time goes by, I can say that. But again, he's an inspiration to us for all he does. But importantly for me, that was a reflection time in my career where for the first time, I left Rochester as a molecular animal investigator and transitioned to more of a medical genetics translational person. So when I came back, I realized that now I have to build a program from the very scratch, because very little they were there available, except for patients who did trust our work in the space. I had the pleasure to work with Francis Collins back then, who taught me a number of things, which I'll come back to my talk. And I realized, as you all do, that for every disease, but as we speak about PSC, every patient is different and at the same time the same. So every patient with PSC has something unique about them, but at the same time, as a group, they have a specific disease that we have to go after to better understand it. So despite this summer, I have to tell you about snowflakes, and this is my best way to depict what I'm saying about patients are the same, but at the same time different. And we like to better understand this difference so we can make better and more tailored therapies down the road. We also realize from the very beginning that there is an interaction between the genetics and the environment. This diagram highlights that for every disease we have, and we're talking about PSC right now, there is always an interaction between what we were born with and what we're exposed to. And we realize that there are few diseases, and probably few people who have PSC, very few, they have a purely genetic element that explains their disease at the very left on my slide. And then there is probably something that is purely environmental that causes this particular disease. But in reality, the vast majority of patients with PSC, they belong in the middle. So they have a genetic profile that's permissive. When they're exposed to some particular agent, which we scratch the area of this knowledge, they develop this disease over time. And so for us, and for me personally, to understand this interaction is crucial for what we can do to bring new knowledge for this disease. So what my laboratory is doing for the last now 20 years almost is to understand the causes of PSC, to understand can we predict PSC progression, and how can we forecast PSC outcomes so that we can understand better how to treat our patients. And going back to Dr. Collins and what I learned from him when I joined his laboratory, I found that he believed strongly that for every condition, including for trauma, there is a genetic component. I think there is value to this. But more importantly, he taught me another thing, without saying this directly, but looking at his own work and what he was developing, that a key element to study a disease is to develop a biobank. And this is quite true, I think, in many different ways. Part of our success, a significant portion of our success, is because we collect biospecimens in an organized fashion, and then we bring our ideas to study those biospecimens. And this is a significant task. It takes a lot of effort, and it's an art and a science, because we have to think about the patient, the interactions, get their trust to provide biospecimens. We create an array of samples. For every single blood you provide, we create almost 15 separate tubes that we have to organize in a way that we can retrieve them as we need them. We had make this decision some time ago that we're not going necessarily to go for newly diagnosed patients, but we're going to go for patients who have already the disease, because if we have just to recruit new cases of patients newly diagnosed, it will take us 15 years to find outcomes of the disease, because this is clearly a slowly progressive disease, as we all know. So this is a retro and a prospective collection, and actually more recently, because of more support, we now ask to have repeat biospecimens so we can see what happens over time for our participants in our studies, which again will give us tremendous opportunities to learn more about the disease. At the same time, we know that the organ of damage is the liver. We don't have access to this. We go peripherally. We collect blood and stool and urine to understand the liver. That's the challenge we're facing. And we're trying to find ways to improve that. But again, as we know, there is no way we can do liver biopsies for research purposes unless there is a clinical indication. And so that's one of the challenges we're facing. At the same time, as Ricky said, about 10 years ago, because of the support we're getting from groups like you, we were putting together a major grant, which I decided to put this name Progress, and it's a mouthful. It's PSC Resource of Genetic Risk, Environment, and Synergy Studies. And as I said, environment was always on our radar screen as far as causing the disease or contributing to the disease, but we didn't have the means at that time to fully study it. It was a great effort that lasted for 6 years. We recruited patients from Mayo and from other medical centers, as you see here, and created the basis of the beginning of our biobank and repository. And every single star on this slide represents a single patient from different areas. And actually, I could say that we have patients from Alaska and from Hawaii, so every single state, although the majority of them, they come from the upper Midwest, as it's the case having the Mayo Clinic in this location. The specific aims of this Progress grant was kind of very simple, was first to understand the risk, the genetic risk that predisposed to PSC, to understand the genetic correlation between IBD, inflammatory bowel disease, and PSC, and then to understand environmental exposures or environmental risks that may predispose to disease. And as I said, for what we do in research for medicine, it depends on the tools we have available. So back then, almost 10 years ago, the tool that we had available is to have DNA microarrays for genetic studies. We started to have platforms or microarrays to study 500 single nucleotide polymorphisms. In the very beginning, we end up with 5 million a few years later. And we use the most latest microarray or chip at that time to screen and to assess about 5,000 patients with PSC, collaborating with 3 groups, our group from the United States, the group from UK, and the group from Norway, to be able to massively address this issue of the genetics in PSC. And also, we took about 20,000 healthy controls that were available in another platform to reach to the point that now we know about 20 genetic loci that contribute to the pathogenesis of this disease. This is the slide that tries to summarize all these findings. For the most part, we know that there are 4 genetic variants in 4 genes, shown here at the very top within the green circle, that predispose to the development of primary sclerosing cholangitis. There are 4 ones listed here that they are present in PSC patients and patients with ulcerative colitis. And then we have 1 that is present between patients who have Crohn's disease and PSC. So there are also 10 of them that are common to all 3 diseases, and that's an area that we try to do more work to better understand these relationships. So clearly, what we know from these experiments and the lessons we arrived to is that there are 20 risk loci that predispose to patients to develop disease. Each one has a very small effect. It makes someone more susceptible by 10 to 50%. And we realized, and we knew that, some of them are present in healthy individuals that will never develop disease. And we struggle to say why they have the genes, but they don't develop the disease. We have some explanations, but that's an area of active research. We also know that the immune system is probably the most significant area of our human genome that contributes to this disease, as in many other autoimmune diseases. And we found that genetically, ulcerative colitis looks more close to PSC than Crohn's disease. We knew this as clinicians. We believe in this clinical information that we gather from our patients, but this was a good way for us to reassure ourselves that what we observed in the clinic, that was the case genetically. So what this progress means? It means that we now, for the first time, have a very good understanding of the genetic architecture of this disease. And this information, this tremendous information, can help us to move forward, because we now like to interact this data, the genetic data, with other omics data to learn more about what causes and what contributes to the progression of this disease. So it's information that will stay with us forever. For every single individual of those 4,000 that we have genotyped one way or the other, we have about 30 million genetic markers across of the genome. This is huge data. This is big data that will stay with us because our genomes will never change for a lifetime. Can we use this information in the clinic? Unfortunately not. It's more for research. As I said, there are a number of healthy individuals who have some of those variants and they never develop disease. So we cannot use this information for a diagnostic test, for example. For the same reason, we cannot screen family members of patients with PSC, because there is no evidence that these genetic markers can help us make this distinction the same way we do for BRCA testing or for other more familial conditions. What are the next steps? That will be the second part of my talk. But again, this information we gather from the genomics, it's here to stay, and I think it gives us a better idea of how specific systems like the immune system interacts with the environment, hopefully, to develop this disease. One of the goals of this project was also to better understand the environment, but at that time, 10 years ago, the only reasonable thing we had in our hands were questionnaires. And I thank you, many of you, who have participated to fill these 35 pages of a questionnaire about exposures. It was a lot of work on your part, a lot of work on our part, but we were able finally to publish this paper where we have 1,000 people with primary sclerosing cholangitis, compared them to about 700 without the disease, to find very little, actually. We found that there is some difference in the diet. We found that people with PSC don't smoke so much. But there was nothing striking about environmental exposures. Part of this is because as individuals, we don't remember how much we have been exposed to what things we live and be around every day. And that's part of the limitation of those methodologies, and that's why we have to think differently about understanding the environment and the impact it may have, or it has, I would say, on this disease. And so I think Francis said to me that in disease, genetics loads the gun and then the environment pulls the trigger. And our quest right now is to understand this interaction, knowing that we have done a fair amount of work in the genetic space. We now know that if you take the liability for PSC, about 20%, 25% for others, it's about the genetics. The rest is our exposures. And so we have to start thinking differently, think out of the box, see how we can use new data, new technology to better understand our environmental exposures. So we need another toolbox. And it's good that this toolbox is available. One of the elements we're working actively right now, it's called the Exposomics or metabolomics, and there are methodologies in place like mass spec, where now from a single drop of blood, you can study up to 1 million chemicals. We don't know what those do for the most part, and many of them are unknown elements, but I think they do something. And so that's our next frontier for what we're doing. And we wrote another grant about a year ago where we're trying to understand the interaction of the multiple omics, including the exposures, the metabolomics, the epigenomics, and the likes, along with the genomics, using artificial intelligence. And this is a major grant from the NIDDK that we were able to put together. And this includes 5 groups. We are the group organizing the whole effort. We have our colleagues from Columbia University and Emory University, where they do all the exposomics, metabolomics work, phenomenal work in that space. We have our great friend and collaborator, Tom Carlson from University of Ulster, who helps us with biospecimens and stool specimens from his own cohort to focus on specific areas of this interaction. And for the first time, we are working now with the mechanical engineers, computer scientists to help us understand this data, because we're creating massive amount of data that we were not in a position to understand without having support from people like the University of Illinois and Dr. Ravi Iyer and his group, where we have supercomputing capacities to make this possible. I never thought 10 years ago or 5 years ago that 30% of my research group will be engineers looking into data and trying to make connections. So what is this grant is all about, for which we're very excited? We like to understand the signatures of PSC in 2 tissues. As I said, we cannot do the liver for a number of reasons, but we have a lot of blood and we have a lot of stool, and we like your support by giving us more biospecimens, because we have the funds to run this analysis. And so we like to go to those 2 spaces, blood and stool, as I saw here, to study a number of platforms to generate about 7 to 80 million points of data for every single patient. And you multiply this by 4,000 times. This is the amount of data we're going to get from blood and stool. And then use artificial intelligence to see what this means for the disease causation, for the disease outcome, for the disease predisposition, and all these questions we pose. As I said earlier, it's probably this interaction between the genes and the environment that contributes to disease. We know from past work that 10 to 40% of the genetic elements are crucial to contribute significantly to the disease, but the rest is unknown. And we think this interaction is probably where we have to go. We call the genes the genome. We call the exposures the exposome. It's the parallel term that Christopher Wilde came to coin about 15 years ago. And this new concept is about all the life course environmental exposures that an individual has in a lifetime, from conception to wherever they provide a specimen. Now, we realize that not all exposures stay with us all the time, but a great majority do, and we'll be surprised how much we can find by doing this type of work. So we embarked on this effort to measure what we thought the unmeasurable a few years ago, and we said there are two elements. There is the biological dose. This is what an individual is exposed, and then there is the biological effect. What is the effect of this exposure to our human body? The first one, we call it the exposomics, and this is usually chemicals or substances we find in the environment. The biological effect, there are many variables, many platforms. We didn't have enough funds at that time to study all of those. We will, as we move forward, we call it metabolomics. And the goals of the study was to say, are there particular exposures or metabolic profiles that are present in patients with PSC compared to control? And is there any interaction between those two elements as we move forward? Now pay attention, we studied just 80 people with PSC, of which 40 had IBD and PSC and 40 did not, because we want to know whether the IBD has a contribution, and then 40 control. So an experiment of about 120 people, not many, but specimens that were collected almost 15 years ago at the very beginning. All of them, they had early stage disease. What we found by doing this elegant experiment and applied this plasma to those two platforms, we found several thousand of measurable chemicals, of which we know some of them, and we confirm some of them. So there is plenty of work for us to analyze even from this 120. Not only that, we have now available software to see how we connect the dots. And we know, for example, here, there is a particular exposure, as you can see at the very right of this slide, that creates a lot of networks about metabolites, all those small triangles here. There are some exposures that they are present, but they don't affect any metabolic pathways, and there's something in between. So this is novel information. Is this something that contributes to PSC? I don't know. I'm just, thinking loudly here. But as we do this work, what was impressive, we found that for PSC, there are 3 classes, and there are some specifics which will come down the road for us. We found a specific fungicide and surfactant and insecticide that they are more prevalent in the PSC population. The odds ratio is how many times this fungicide is present in a patient with PSC compared to a healthy individual. This is a significant number. Now, to put this in perspective, we know that asbestos causes lung cancer. The odds ratio is 4. So this fungicide, it's almost twice the magnitude. Now, I don't say that this fungicide causes PSC. We're not there yet. We have to confirm it. We have to understand this, how it works. Maybe what simply means is the individual patients who have PSC, they don't have an intact liver machinery to metabolize this. But even if this is the case, we're going to understand the defect of this metabolic pathway. More importantly now, we know that there are a number of metabolites like the chenodeoxycholic acid, taurochenodeoxycholic acid, that are very prevalent in the PSC population compared to controls. And now, for the first time also, we're trying to make this connected. So we know that this particular fungicide, as I saw here, it creates a lot of network effects to a number of metabolic pathways shown here on the very right of the picture. So this is novel data, very exciting data that we like to move forward to better understand this introduction. It's possible. We made it. We have a lead to follow, and we go after it. We think that there are risk loci from the genetic standpoint that we have described. We think there are environmental exposures that we now try to understand, and this interaction causes PSC. And we have to think that probably the genetic loci are going to be very rare. The environmental exposures are going to be very common. And they're going to be common exposures for all of us, like household products, pesticides, other things that we come into contact every single day. And it's a contribution of them over days and weeks and months that cause interaction. We're not going to find asbestos. We're not going to find smoking as the risk factor. We knew this because when we did our questionnaire, none of this came up. Actually, even for pesticides, nothing came up. We were asking this question. The reason why is that nobody knows what we're exposed to. And that's what I like to resonate here. So we can measure the exposome and the other omes. We have now a unique opportunity to do more work in that space, and we think that with all this network analysis that we bring together with exposomics and metabolomics, we'll find new ways to think about this disease. Now, as I said earlier, we used 120 specimens to run this analysis, and we had some significant findings that are now under review for publication. We're putting forward over the next 4 years 4,000 biospecimens, of which many we have already in possession. We need more from patients to better understand what happens over time, and that's what we'll focus in the next year or two. This will give us tremendous opportunity to understand this, this data, and will make discoveries of many of the unknown features that are present, but we don't know what it is. And this information will generate hypotheses that many laboratories across the globe can then go back and study what this means to the particular entities we make discoveries on. A major goal of this grant is to develop a community resource for everybody to use and share. And actually, a lot of our resources and a lot of our work we're doing is to create all the platforms of the raw data to disseminate this to other investigators, to other institutions, to work on this so we all learn about it. Clearly, what we're doing, we're developing a massive big data for PSC. This is where we go. This is what we have to do, and we should do to understand this disease and trying to bring therapies forward. Now, if I have to, bring the correlation in a pictorial, I'll show you here a puzzle of a picture. This is about 300 pieces. Imagine we are going to have 300 billion pieces of data from all these 4,000 patients. Don, do you know what this picture is? Wrong. So this is The Simpsons. Okay, you don't know what you find until you do the analysis, but this is the amount of work we had ahead of us to make this journey to find new solutions for patients with PSC. I like to thank all of you for coming to Rochester. I like to thank all of you who trusted your biospecimens to us to do studies. Without your support, none of this work will be possible, believe me. I'd like to thank our benefactors, the Carlos family and the Haldoran Family Foundation, for supporting us, the PSC partners for all the work we have done over the years, our collaborators from Oslo, University of Illinois, Mount Sinai, Emory, and Columbia. I'm very blessed to work with great people in my own laboratory that we have here in this photo. In the very early years, I was doing everything, collecting biospecimens, writing protocols. I do very little of many things. Actually, they keep me out of the lab because I think I create more problems than solving problems. And so I do the best by thinking forward and thinking loudly and thinking broadly about this disease. And that's the main thing they let me do. And it seems it's successful so far. So thank you again for your attendance, and we'll be happy to answer any questions.
All right. Thank you, Kostas. Excellent talk. Our next speaker is Dr. Greg Gores, who many of you know, professor of medicine and executive dean for research at Mayo Clinic. Really a world-renowned physician scientist with vast expertise, but more than anything, I think Greg has simply devoted the majority of his adult life to understanding this disease and finding new treatments. So we're lucky to have him here today. He's going to talk about animal models in PSC.
Thanks, Dr. Huebert, for your generous introduction. And it's a real privilege to be with you this morning. As Kostas said, you are our motivation and our inspiration to keep evaluating this disease and find a cure. Our CEO has challenged the whole institution to think about cure. And it's an aspirational goal for many diseases, but I think the focus is correct, and that is our goal. I personally have been seeing patients with PSC since 1985 when I was with Dr. LaRusso, and so I understand the complexities of the disease, your concerns, fears, and hopes. So Costas, let me just tell you a little bit about the team science, convergent science. We're so fortunate to have at Mayo. So I think Costas gave you a peek into the future of research. I'm going to take you maybe a little bit more old school and look at the reductionistic approach. But every Thursday morning, he and I and Dr. LaRusso, Dr. Huebert, and Dr. Eaton sit in a room for an hour and think about what we're doing, where we're going, and what we hope to accomplish in the next couple of weeks in a very dedicated manner. So I'm going to talk a little bit about animal models, probably many which you haven't really thought about. And so we need animal models to understand how the disease occurs and what therapies may be important. So almost all therapies require some testing in an animal model. Okay? The whole idea that you could use artificial intelligence, search the literature, look at the exposome, look at the genetics, pull what we know about information, and then develop a drug from a computer-based approach doesn't work because biology is so complex. So the ideal model will recapitulate the disease. It'll allow us to look at genetic manipulation, what's the effect of that specific gene. We can do pharmacologic studies, and they're also required for FDA approval processes. The cons of animal models is that even though we share a lot of gene similarity with animals, they're not identical genes. And the disease processes in animal and humans can be strikingly different. We have very few large animal models of disease simply due to expense. Drug metabolism is different, so the mouse metabolizes drugs 10 times faster than humans do in a much different way. And models are time limited. We study animal models in days, weeks, hours, months. Humans have diseases for years, decades. So the ideal animal model of PSC would probably, from my perspective, would have inflammatory bowel disease, would have large bile duct strictures, it would have progression over time to cirrhosis, it would predispose the animal to development of biliary tract tumors, and we do not have the ideal animal model today. We're still thinking about it, and hopefully some of the work from the large genetic studies and environmental exposures may inform animal models, but today we don't have that. Our animal models of IBD are also surprisingly lacking. We have toxic injury of the intestinal lining. We have genetic models of a disease such as interleukin-10 deficiency, and there are humans with defects in IL-10 who have inflammatory bowel disease. There are a variety of manipulations you can do in the mouse, primarily genetic, adoptive transfer or something. They have IBD, but none of these animal models have PSC. So we cannot recapitulate backwards the association in the disease. So what are we doing? What's the current state of what we do, and what have we learned to date? So we have both acute models of PSC in mice, which we have developed here. We have a genetic model, which I'll get into in detail, that is commonly used in the literature, and then we're beginning to think about biliary tract malignancies. So I won't go into a lot of the details, but we did develop an acute model of sclerosing cholangitis in the mouse with help from a grant from you all. And basically what we did was we developed a drug into the bile ducts, which affects signaling in the bile ducts, and we were able to get a sclerosing cholangitis picture. So I will simply say that in the mouse, we can inject things directly into the gallbladder. We can leave the drug there for a while with a clamp. Then we can release it, and then we can look over time. And really interesting, I will just highlight a couple of things from this model. We got a histology of the bile ducts that looks identical to PSC with onion skinning, fibrosis around the bile duct. And we had elevations of the ALT and alkaline phosphatase and bile acids, and the mice actually became jaundiced acutely. And I will note two things. One is we were able to show for the first time that there was this increase in a type of cell, a white blood cell in the body, termed a macrophage, which people usually thought just fought infections. And we were actually able to show that this cell type was recruited to the bile ducts and caused injury of the bile ducts. And this was sort of the first time that this type of white blood cell was shown to be contribute to PS, to a sclerosing cholangitis effect. We were also able to get strictures in the bile duct in the mouse. This is a cholangiogram of a mouse, and we could get strictures. The interesting part is 2 weeks later, these mice were normal. We did this once, and they were normal. And it shows that all these changes that we attribute to advanced disease, onion skinning, fibrosis, and scarring and things, are actually pretty reversible. And if we can learn more about this disease, I think we will be able to understand the reversibility of the process. And so we've been thinking about how these macrophages accentuate the disease process and causes sort of onion skinning fibrosis in the mouse, and then I'll come back to that story. The other model, which is extremely popular across the world, is this mouse that lacks a gene involved in forming bile and moving things into bile. It's a defect in, if you will, in phospholipid transport. It causes a PSC-like cholangiopathy in the mouse. And it's interesting in that the gene sits on this side of the hepatocyte and pushes these phospholipids into bile. And there are certainly humans who have this disease defect, but they don't have PSC. So then again, this sort of highlights the differences between mice and men. The mice look like they have PSC. Humans don't. What humans get is a biliary tract injury of small bile ducts that can progress over time. They get the so-called cholestasis of pregnancy. They can be predisposed to stones in their bile, these kind of defects. So it's a common in the liver practice to see these patients, but they don't have PSC. The mouse does. And this just shows the details of the bile ducts in this mouse model. So it looks a lot like, this is a cholangiogram from a paper Michael Trauner wrote several years ago. And this shows a lot of stricturing and other abnormalities of the bile duct as compared to a normal mouse bile duct. We also showed that these macrophages are quite prevalent around the bile ducts in this more chronic injury. And so these kind of concepts, why are they important? Because they lead to this sort of thinking that maybe if you block the monocytes or these macrophages going to the bile ducts, it would be beneficial in the disease. And so this is where animal models can be very helpful. And in fact, there are such drugs that block monocyte macrophage recruitment to tissues. There's actually been a small clinical trial of this drug in a small number of patients with PSC over a small time frame. And according to websites, the trial has completed enrolling. I have not yet seen any data from such a trial, but then we could have a long discussion about what the legitimate endpoints are for a short-term trial in PSC. In the mouse, anyway, these little brown spots are these cells which are sort of attacking bile ducts in the region. And when we give this drug, CVC, for the sake of an abbreviation, you can see we prevent the recruitment of these cells to the bile duct area, and something that could be potentially recapitulated in man and would be helpful. So this would be, you can imagine a bile duct has an environmental insult in a genetic background. A diagnosis is made. You could then administer a drug which prevented these monocytes from going to this area and prevent this sort of scarring around bile ducts. So this is the advantage of an animal model. This is some of the information that we're currently generating. Also wanted to take, this is a busy slide, and I won't go into all the details of the slide, but I would point another advantage of these animal models. So Dr. Huebert and I and others are very interested in some of these, what happens when a bile duct is injured. The liver has a regenerative response. So regenerative responses are kind of like wound healing responses, right? You want to heal the tissue. Part of the healing response is to make some scarring, so you can seal the defects. And what happens in human disease is that the healing response never stops, or the regeneration response never stops. And that sort of unrelenting, chronic reparative process then sort of causes scarring of the liver, and you end up with cirrhosis. And one of these cells that participates in this response is called the ductular reaction, and all these sort of brown cells here and out here would be examples of the ductular reaction in a mouse model. But similar cells, identical cells, exist in human disease, including PSC. And we've started to say, well, what if we just intentionally, purposefully killed those bad cells? Could we reduce the fibrosis? And this is some ongoing work in our lab, but I will just say that here you can see all these are cells that are scarring the tissue. We purposefully have killed these ductular reactive cells with a drug, and now you don't see much fibrosis here. And if we quantitate fibrosis, it drops down carefully. So I would just say we're looking at different ways then to prevent the scarring and to prevent recruitment of inflammatory cells to the liver that can really only be done in animal models with a purposeful eye towards therapy. Finally, as many of you know, we're very interested in biliary tract neoplasia. Why does that occur in this disease? And I would just say that we're also looking at ways to inject potential genes into the bile ducts that can be associated with cancer, and we're also focused on this complication of PSC. So I really just want to end and say animal models of PSC are important to understanding the mechanisms of disease and to test preclinical therapies. This is essential. I think the PSC Partners Seeking a Cure has helped support this kind of really critical research, often by very junior people who needed to be attracted into the field. And so kudos to the foundation. And then I just thanks for everything you do to raise awareness for PSC and support research relevant to this disease. I can remember several years ago, I was a keynote speaker at Mayo Clinic, Florida. One of the participants reminded me of that yesterday. And I can remember sitting at a table and with these young people in one of the breakout sessions and really asking very emotionally, what can we do to help? And I think what you can do to help is what you're doing. Help provide blood samples and other samples so we can understand the genetics and the exposome. Participate in clinical trials where it's reasonable and appropriate, and help support research in this activity. And I do really think that we will reach the goal of a cure. Thank you.
Thank you, Greg. Our last speaker for this session will be Nickie Francisco Ziller, who's a dietitian, works in our liver transplant center, seen many, many patients with PSC in the outpatient setting and in the inpatient setting. And I've already heard a lot of really great questions about diet from the attendees, so I think this talk will be of great interest.
Thank you, Dr. Huebert. I'd like to thank all of you for coming today to hear about nutrition and how you can use that as a tool to help yourself maintain good health throughout your journey with PSC. So thank you. Our objectives here today to talk about healthy nutrition while living with PSC. We'll address nutrition briefly for those with IBD. And if time, we'll talk about food safety. The first slide here is a busy slide, but I just wanted to start out reminding everyone, as you know, there are many roles that the liver has that are very important, and you find out what they are as you go through the process. But a few things really relate specifically to nutrition. The liver stores various vitamins and minerals. It stores energy as glycogen, and that glycogen plays a role in glucose management. The liver synthesizes proteins. It produces bile, and as you know, bile plays a role in the absorption of fats after we eat our foods. And the liver filters harmful substances. So I'll be addressing some of these as we talk. Potential nutritional issues that relate specifically to PSC. We're going to go through each one of these separate, but the absorption of foods and nutrients, vitamins and minerals, bone health, lipids, for example, cholesterol or triglyceride levels, and your weight. So first of all, talking about malabsorption. If a patient has an elevated bilirubin, then less bile is getting into their intestinal tract, and one of bile's roles is to help us absorb fat. A person who isn't absorbing fat very well might notice that they have more stool output or lighter colored stools. The stool might float or be difficult to flush. These would be indicators that you're not actually absorbing all the fat in your diet and that you're losing some of those nutrients and calories. Some of the nutrients that need fat for absorption are the fat-soluble vitamins A, D, and E. They are the most likely vitamins to be low when we check the blood in patients with PSC who have an elevated bilirubin. Vitamins and minerals in general are stored, some of them are stored in the liver, some are activated in the liver into their usable forms. People with PSC are at increased risk for vitamin deficiencies, and like I said, specifically the fat-soluble deficiencies. These can be checked with a simple blood test, and then depending on which ones are low and how low they are, your physician may recommend supplementation for those. Vitamin E is an antioxidant. Oh, I'm sorry, let's start at the top, shall we? Vitamin A is a vitamin that affects vision, most specifically night vision. Vitamin D can be beneficial for bone health. There's also many other things that vitamin D is being researched for currently. And vitamin E is an antioxidant that affects the health of blood, brain, and skin. Now a bit on bone health. You may know that osteopenia and osteoporosis develop to some degree in most patients with cirrhosis, specifically cholestatic diseases such as PSC. I believe you had a talk earlier in the session about bone health, so you may have already be familiar with that. People with PSC should always be screened for their bone loss. The reasons that they might lose bone is multifactorial, but it includes the fat malabsorption that I referred to earlier. Also, it includes decreased muscle mass. Sometimes as disease progresses, people are less active, and then they have less muscle mass, and decreased activity in general. If low bone mass is identified, it would be beneficial for you as a patient to choose foods that are high in calcium and high in vitamin D, such as dairy products, milk, and yogurts that are fortified with vitamin D. Your physician may also recommend supplementation. Weight-bearing activities would help your bones as well, so maybe very light weights, brisk walking, and things like that will help keep your bones strong. So the next section is about lipids. This is referring to cholesterol levels primarily. Bile is involved with the excretion of cholesterol. So if bilirubin is elevated in your bloodstream, you might find that your cholesterol is also elevated. And I don't want you to be alarmed by that. That's a common occurrence if the bilirubin is high. Research has shown that there is a correlation here. But studies that have looked at patients with PSC or the correlating disease of PBC have shown that elevated cholesterol seen in the situation of cholestasis does not carry the same cardiac risk that it would if the patient did not have a high bilirubin or cholestasis. So really, a low-fat diet is not indicated in this scenario. Okay. Now I wanted to talk about weight as an issue. Basically, we all fall into three categories. We either weigh too much, we weigh too little, or we're just right. Pretty much summarizes the weight thing. I listed on the side the BMI criteria for weight, and many of you may be familiar with BMI, which stands for Body Mass Index. A normal, healthy BMI is 20 to 25 or 20 to 26, and that's where your goal would be to fall. An excessive BMI indicating that someone is overweight or obese would be a BMI greater than 30. And a weight that is low would be a BMI that's below 19 or 20. There are calculators online for how to calculate your own personal BMI if you would like to know that number. Know that it's certainly not a perfect tool, as none of them are. BMI is affected by fluid retention, so if a patient has ascites or edema, their BMI is going to appear to be higher than it truly is. So the tool is only as good as it can be, but it's something we can use. The focus, no matter what category you fall in, is to preserve your muscle mass and strength throughout the process. So we're going to go through each weight category a little bit. So being of a normal weight, your goals will be, like most people, to choose healthful foods overall and a wide variety in your diet with 5 servings of fruits and veggies every day. Choosing lean sources of protein, healthy types of fat, and when you eat carbohydrates, choosing the whole grains over the white or refined products. Choosing low-fat dairy products like skim or 1% milk would be better than the whole milks. And then regular daily exercise. If you're in the overweight group, your goal would be to get your body mass index below 30, but in a healthful manner. So we don't want you to skip meals in that effort to lose weight, and we don't want you to implement a fad diet that might eliminate a food group or an entire macronutrient from your diet. So you'd want to be sensible about how you do this. Focusing instead on appropriate portion sizes, healthy choices, and exercise. We don't typically use a weight loss medication in this patient population. What I don't want you to do is skimp on protein, to save on calories. So protein is very important, that you continue to get that protein source at every meal. People who are underweight or have had unintentional weight loss, I want to be very concerned about that and to actively try to stop that from that trajectory. So you would try to eat smaller, more frequent meals and snacks. Thinking about how can I increase the caloric density of my foods, which kind of goes against how we've been thinking about our nutrition our whole life, trying to eat lower calorie things. But in this scenario, it's important to look at what you're choosing to eat and think about how can I eat the same quantity of food but have it be more calories. So for example, a piece of toast. You might have put diet jam on before because you were trying to save calories. And now you might think about putting butter or margarine and peanut butter and maybe jam too, but how can you make the same piece of toast fill you up in the same way but give you more nutrients and more calories for that same amount of food? Again, having a protein source at every meal is very important. Some people may need to use nutritional supplement beverages. Examples would be like Boost or Ensure. I'm just throwing those names out because they're the most familiar for most people. They're available at grocery stores, pharmacies, Target, Walmart type places, and online. If you're not sure which one is right for you, you can ask to meet with a dietitian and see. When you go to the aisles of these nutritional supplement sections of the store, there are so many now that it really is pretty overwhelming. Are you supposed to get the plus version or the high protein version or the immune health version? There's many, many that are there. If you try them and you don't care for them, it would be good to try a different kind, a different brand, or a different flavor, and give it more than one shot. Okay. Regular daily exercise is also very important for the underweight or the people who have lost weight. Sometimes people have expressed to me that they're concerned if they exercise, they're going to burn up all the calories that we've been working to get them to take in. But I want you to think of it more as utilizing the calories you're trying to take to maintain or build strength in yourself. So we still want that activity to occur. This slide just summarizes some of the eating difficulties that some patients with PSC can have. It includes changes in appetite, fatigue or decreased activity, malabsorption, the presence of fluid retention, as in ascites or edema, changes in bowel habits or a flare-up of the IBD. Many people become full quicker than they used to, so they're eating smaller amounts of food and they feel full. Various medications can give our patients side effects, such as antibiotics that change food's taste or lactulose that gives people diarrhea. There's various medications out there that cause some nutritional side effects. As disease progresses, some patients can have recurrent episodes of cholangitis where they either need to be in the hospital or they need to have ERCPs, and that holds up a day or two of eating, and that all contributes to having less nutrition in their body. I'm going to slip into a couple slides on IBD now. So don't get angry with me for this slide, but there is no one-size-fits-all diet for IBD. As much as everybody wishes there was, including your caregivers, there is not one defined diet that works for everyone. Individual people might find that something works well for them. For example, some do better off lactose. Some limit their gluten and feel better because of that. Some patients just simply do not tolerate high fiber foods or high amounts of fresh fruits and veggies. And others find that gas-forming foods bother them. What I don't want people to do is start to restrict so many foods in their diet that their diet becomes very limited. So some people keep food diaries and they try to keep track of what triggers them to not feel well. I just don't want everything in your diet to get eliminated because of that. Sometimes a flare-up was going to occur and it didn't matter that you just ate corn or not. It was just going to happen. Suggestions for those with IBD, though, would be to chew your foods well. Think about that initial process of digestion. It starts in the mouth. And by chewing well and not gulping down your food, you're providing less work for your intestinal tracts to do. Eat a wide variety of foods as is allowed within your diet restrictions. Drink adequate amounts of fluids. That helps push things through and keep your bowel habits more normal. Many people will do better by eating smaller, more frequent meals and snacks. So again, you're giving your intestinal tract less work to do at one time. So it might be able to be more efficient with smaller amounts more frequently. During a flare-up, it certainly might be a good idea to back off on the fiber foods that we know are good and healthy for us. But if you're having a flare-up, you might just back off on those. If you have an ileostomy, that presents a little bit different of an issue sometimes. So people might find that, especially in the first few months after the ostomy has been placed, maybe after they drink a higher sugar-containing liquid, they can have a higher ileostomy output. So for example, a juice or a pop might go through the system quite quickly and increase that ileostomy output, which can lead to dehydration. That's pretty individual for people, and it usually should get better with time, but it is something to keep your eye on. If you drink juice and your bag fills up 20 minutes later, you might want to dilute that juice, half water, half juice, or not drink pop, in my opinion, but dilute other sugared beverages, half and half. If gluten is an issue for you, like you strongly feel that gluten-containing foods bother you, you may actually want to take it a step further and ask your physician to do more testing. You want to know that you do or do not have celiac disease. It's important to know whether it's just an intolerance that's easy to manage or truly celiac disease. There's a couple things. Turmeric is kind of one of the hot items right now, and then other probiotic type foods that are being researched for people that have IBD. So hopefully we'll hear more on those topics as time goes on. A couple of slides on complementary medicine. So in general, if you want to start something, you should speak to your pharmacist or dietitian or physician prior to doing that. And I think everybody probably knows that herbal supplements are not FDA approved. Therefore, we have much less data on the safety and efficacy of those products because the trials don't have to be done. And specifically in liver disease, we do not have much information in that scenario. And herbal supplements have to be filtered by the liver, like all things. So we are more hesitant in this scenario to agree to herbal supplement use. One key point is the last point on this slide is that natural is not the same thing as safe. They are two completely separate words, but they're used often when someone who is maybe trying to sell you a specific product will tell you that it's okay because it's all natural. Well, there's poisonous mushrooms that are all natural, and there's arsenic in our soil that's natural, neither of which I recommend that you ingest. So it's the same idea. Natural and safe are different. It's certainly sensible for most of us to be taking a daily multivitamin that contains 100% of the RDA of nutrients. We do not need mega doses of things. We don't need a multivitamin that gives us 10 times of the recommended daily doses, just a standard multivitamin. Your physician may advise you to take specific vitamins and minerals. As we discussed, it might most likely be vitamin A or D, would be probably the two most likely. Be very wary of misinformation. There's a lot of it there. You can find it very easily on the internet or even from your neighbor who sells something. So just be wary that we have the research that we have in liver disease. It would be specific, and often it does not include the newest, latest thing because we would need a whole bunch of people with PSC to agree to take that item. There's studies that may not ever be done. Nutritional supplements might be advised by your dietitian or your physician. They are not magical, but they are nice because they are pre-made. They have the right amount of nutrients for most of us. And usually they're a concentrated form of calories and protein that's typically easy for us to get down. So for example, we might have a person who's very tired out and fatigued and doesn't want to go make lunch for themselves because they're not very hungry and they're tired. They could at least drink a nutritional supplement drink to get through that time period. The last one just comments on milk thistle. The only reason I'm bringing it up is because it's commonly recommended for people, or you might hear about that one. That particular product has been tested and considered safe. That's the important thing to know. It has not been found to be particularly beneficial, but I think the key thing to know is that if you chose to try it, it should not harm you. A few other things that are coming up and I think promising in the setting of liver disease are omega-3 fatty acids. Most of you are probably familiar with omega-3s. They have an anti-inflammatory effect for us, and so that's a good thing for both PSC and IBD. The omega-3s are typically in fattier fishes like salmon and tuna, also in trout. Omega-3s are in some of the nuts, mostly high in walnuts and almonds. They're in flaxseed and chia seeds, which are easy things to add to your cereal in the morning or your granola. Another one you may or may not have heard of is resveratrol, and that particular product is also high in antioxidants, and it's in some of the fruits I have listed here, like grapes, blueberries, cranberries, peanuts, tea, and cocoa. Specifically cocoa, like that's 50% or more. So that product is also in red wine, which is not necessarily the recommendation for this population, but that's where the grapes come in. You have to eat it as grapes. So probiotics, as you probably know, are live bacterial that can help with intestinal health, that can help with immune health. There's a lot of potential benefits in the probiotic field. At this time, we would generally recommend that you use that through foods, as in live culture containing yogurt or kefir. If your physician specifically recommends probiotics for you, that's great. I would just talk to them first. If someone has ongoing infectious issues or they're immediately post-transplant, we may discourage the use of probiotics at that time. In pill form, I'm sorry, not in food form. You can have it in food form. The last one, coffee. I am just bringing this up because for those of you coffee drinkers out there like myself, you do not have to feel guilty about drinking coffee in the setting of liver disease. So yeah, it has been studied. Moderation, okay, moderation. It has been studied and shown to not be harmful in any way to liver cells and potentially even a benefit. And that would be about 3 cups of coffee a day. Regular or decaf have both shown this benefit. So again, just not something that I would start to do if you don't already drink coffee, but if you do, enjoy. I have 3 food safety slides. Do I have time, Dr. Huebert? Or I can...
A few more minutes.
These slides are on food safety, and they're in your binder, in your folders or online. And so review them as you wish, but just to run over this quickly. Washing all produce before eating and peeling is very important, and I mean all produce. Bagged lettuce, cantaloupe, things that we might not have typically washed. Running or scrubbing under running water. Egg yolk should be cooked until the white and yolk are firm, which is kind of a bummer. The other one that I would say is difficult for most people is the deli meat issue. The meat sliced at the grocery store delis is higher in listeria content than the FDA would like, and the recommendation is that that's actually heated before you eat it. Things to avoid: raw sprouts, eating raw or undercooked fish or meat. We want you not to have unpasteurized dairy products, so the milk that you purchase at the store is all pasteurized, but if your neighbor, milked their goat and brought you some, that would be unpasteurized milk that you should not ingest. You wouldn't believe what I hear. So be cautious about eating at buffets or at salad bars. They are definitely a setup for more bacteria being in the food that is sitting out and multiple people coming along and helping themselves. Washing your hands before handling food, washing cutting boards before you prep the food, and washing all produce. Being careful about cross-contamination so that you're not putting like your raw hamburger just straight on your refrigerator shelf. There should be a plate under it. Keeping those things separate from each other. Cooking your meat and fish, cooking your egg whites. It's safest to use a meat thermometer, especially if you're grilling, to know that the chicken that looks done on the outside is actually at the 160 that it needs to be on the inside. And thawing foods in the refrigerator or the microwave, not so much on the countertop all day while you're at work. Okay. The bottom of this slide contains a website that I would encourage you to go to. There's a lot more information on food safety on this website. Last slide. Overall, healthy nutrition for the liver involves working towards and staying at a healthy weight. I didn't discuss this before, but we want you not to skip meals, so eating 3 meals a day and then always a bedtime snack. And this becomes more and more important as disease progresses. So another awesome thing the dietitian told you to eat before bed. How good can it get, right? So that helps minimize muscle wasting overnight in the setting of liver disease. Choose healthy fats. Avoid adding salt to your food, which can contribute to that fluid retention. Having a protein source at every meal. Limiting or avoiding alcohol. Be active most days. And in general, avoiding herbal supplements unless you've approved this with your medical team. And that's what I have for you. Thank you.
Thank you, Nickie. So I'll invite the other 2 speakers up for a panel discussion. If I understand correctly, we have some people who will run questions to the front. Are you there? Can you raise your hands if you're there? Okay, so see those people, give them your questions. We'll try to get to as many as we can. All right, they're rolling in already. What are the, I don't know, this might be a question for Greg or Kostas, but what are the thoughts on the beaded pattern of the bile duct involvement? Certain areas are spared and others are affected. Why is that? Is there any ideas about that in the field?
So let me rephrase the question. The question is, certain areas of the bile duct appear to be more involved in a disease process, and what are the insights that we have there? I would say that, first of all, there's tremendous differences in the cells that line the bile ducts, from the very small bile ducts to the very large bile ducts. So not all bile ducts cells are the same. And so we now know that the cells in the bile ducts participate in the inflammatory reaction, right? They also get agitated and change their normal function of just simply contributing to bile flow, but participate in the inflammation. And so because they're different in different sections of the bile duct, they likely participate differently in that process.
Great. Nickie, this one's for you. If a person with PSC has high cholesterol, should they be on statins even if they have a family history of high cholesterol?
That's a good question. They don't need to follow a low-fat diet unless they have an actual heart history. As far as statins go, I would let one of the physicians answer that.
I think it's our long-standing practice at Mayo not to use statins in people who have liver disease with impaired bile flow. So there's no increased risk, and Kostas will verify this. There's no increased risk of cardiovascular disease from the hypercholesterolemia of PSC. So we do not treat it. On the other hand, from a precision medicine approach, if you have siblings or parents who had cardiovascular disease early in life, you have hypertension, you smoked for 20 years, but very unusual in PSC patients, but then you may want to reconsider. So I would say you have to take into the context of your other risk factors, but as an isolated risk factor, no.
So some themes here, a lot of questions. I don't know how many we'll get to, but we'll try. So on the one hand, fungicides may be somehow causative in the disease. On the other hand, there is no specific diet. I'd be a little confused if I were in the audience. Should these people be turning to organic farming and avoiding pesticides and things? That there's some themes to that effect.
Great questions. We try to understand the landscape of the genetics and the exposures that contribute to the disease. All of us were exposed to chemicals on a daily basis, but we don't develop disease or we don't develop PSC. So we have to understand more about this interaction. Now, at the same time, I think common sense for less chemical exposures, it will be maybe good for every disease or every disease state. So I think this goes along the lines of healthy food, local produce, those things are important. We know that there are about 160 million exposures registered with EPA, 160 million exposures and chemicals, of which about 40 million just about the last 10 years. So there is an explosion of chemical exposure that we have seen, and it appears that all of us, we've been exposed to about 1.5 million, 2 million on our lifetime. We like to understand this interaction. We like to understand over time if there is a progressive accumulation of some of these chemicals or fungicides in our diet. And also, we think that it's not a single high exposure that will make the difference. Actually, we took all the patients that we have registered. We saw where they lived. We tried to find out, is there any cluster of cases along, across the United States? We didn't find anything, which means that the exposures that we're talking about are simple daily exposures that probably accumulate over time to develop, the phenotype of this disease. Again, common sense about diet, wash your food, but I think at the same time we'll make discoveries to see how this will play out down the road. I believe that there are people who have a genetic profile, and probably the immune system will be key there. They have an immune profile that when they're exposed to some particular chemical, which can be innocent for another person, it will trigger an effect that will focus on the bile ducts, and maybe that's what it takes. Greg, your thoughts?
Okay. Rapid fire nutrition. Yes, no, or we don't know. Is tea okay?
Tea is good. It's a high antioxidant food.
Venison has a lot of omega-3 or no?
Venison is generally lower in fat than other meats, so I wouldn't say it's a high omega-3, but it's a good, healthy food that you could still enjoy in your diet.
Celery juice.
Wow, that sounds so bad. Sorry. I have never had celery juice. I'm sure it's perfectly good for you. It's not the highest, like it wouldn't be like kale juice or something that would have more nutrients than that, but it would be fine.
A theme is cannabis, so that would be either marijuana products, CBD oil, or hemp protein added to foods.
I would consider CBD oil in the category of complementary medicine that you would need to ask about. Personally, I would discourage that. It's very expensive also if you haven't checked into the price. Hemp protein is fine to use as your protein source. The first was marijuana?
Yeah.
Yeah, I can't really encourage that, so
Good.
sorry. It can be an appetite enhancer, and there is a medication that some people use for that purpose.
But there's also some data that THC can be pro-fibrogenic in the liver.
Okay, so
Maybe they don't
15 to 20% of patients have recurrence of PSC after a transplant. Others do not. Do we have any idea what may influence that?
Can you repeat the question, Bob?
So why do some patients have a recurrence of PSC after a liver transplant and others do not? Do we know? Do we have any ideas?
You could also take that question. But I guess you're the moderator. So the data, I think what the best data that we have is, let's say for the sake of discussion, 20 out of 100 people will develop recurrent PSC by 10 years after transplant. So it's roughly a 20% risk. I think it often resets the clock. It often is very indolent and often doesn't impair one's health for prolonged periods of time. I think it speaks to the fact that there is an immune reaction between the host, you, and the bile ducts that can easily be triggered, and that the genetics of the host liver aren't probably that important in PSC. So it informs biology. There are no really good data to show why some people get it, why some people don't. And finally, I would note that the environment of the liver is much different after transplant because you have sewn up the intestines to the bile ducts, and the role of that sort of bacterial colonization, the new microbiome of the bile ducts in that scenario hasn't really been fully examined.
It's a very complex question. You know, I agree with Dr. Gores every single point he makes. The only thing I would say also from the standpoint of studies, you know, to do genetics, you need thousands of people to reach the outcomes. And so if you have 20% simply of patients transplanted develop this situation of recurrence, there are not so many for genetics to be able to study the phenomenon, and that's the limitation we have.
Does PSC progress slower in patients who had their colons removed, and do we know why or what this connection means?
I'm not convinced that it progresses slower in people who have had their colons removed. We certainly have seen patients who have developed PSC after they've had their colons removed. And we certainly have seen patients who have developed inflammatory bowel disease after they've had a transplant for PSC. So I don't think and the classic teaching is that there's no relationship between the activity of the inflammatory bowel disease and PSC. And so I think that those studies that suggest there may be a relationship, I would say the size effect is quite marginal at best.
Another theme here is agents taken for toenail fungus. I think these people are now terrified that they should not ever have any exposure to any kind of antifungal, and that it may have caused their disease.
As I said, there is a lot of data for us to analyze. We're going into this space. We're going to have 4,000 participants in our studies, and I think many unknowns to understand better. So we'll have answers. We know nail polish, for example, associates with a primary biliary cholangitis. And so we're going to find features down the road that will associate. Then how do you make the association a pathogenesis of the disease? It's another question.
Toenail fungus never killed anybody, so why take something?
This is a question I asked Greg a few weeks ago. So you gave this mouse had PSC, and you gave it something, and the ducts returned back to normal. How did that happen so quickly?
I think the processes are surprisingly reversible. Dr. Hebert and I were just speaking about that this morning. So I think much of what we have assumed to be progressive disease of long duration is in fact a very dynamic process. That there's an ebb and flow every minute of the day in the process, and if you can interrupt the aggressive factors, you can return things to normal reasonably well. I think the only exception to that is in advanced fibrosis, where the collagen is heavily chemically cross-linked, and one can no longer the body has no mechanism to chew it up. But up until that happens, the liver is remarkable. It's reversible and can return to normal.
Some people are wondering how they can contribute their samples. And then some other questions along those lines are you able to mine some of these direct-to-consumer things like 23andMe, Ancestry.com, where there's genetic data that maybe PSC patients have contributed to?
Well, thank you for your interest to participate. We have a booth outside. I think it's on the other side of the building today, I may be mistaken, where you can go and learn about our studies and how to participate. We have phlebotomists on site to participate at your convenience. We can also simply present the study to you, get the consent with you, and we can do the rest of the collection by mailing. So we'll be happy to accommodate if you want to participate by any way. Bob, can you repeat the second question?
So there's probably data out there with 23andMe and Ancestry.com and other sources to mine some of this data.
Yeah, the data is very limited and is very focused. We need more granular data to analyze. There is a lot of information in the genome. What we do, we go after several million of markers for what we have, and what those commercial entities provide, there are a few SNPs, 15, 20, 100, so it's very limited information. It's a good information for what they're going after, but it's very limited, and that's the limitation.
It's much more valuable to the police than it is to Dr. Lazaridis.
Now, one more comment I would like to make beyond that is about about diet and how your help can be crucial. It reminds me, 7 years ago when we had the meeting here in Rochester back in 2012, I was in a room with 10 other people and we're talking about diet and exposures even back then, although we had no idea, whether we will go to that direction in a short period of time. But I asked one question about whether people smoke or drink or have coffee, and I found that none of those 10 patients ever had coffee. And I went to Ricky and I said the next moment, Ricky, we're going to develop a study to study this observation. Actually, we did, and in the Norwegians, they did at the same time, and that's where we found that people with PSC, they don't like to drink coffee, and this associates with disease in a negative way. So if you want to drink coffee and you drink coffee, please do. I don't know how this creates a protective mechanism, but that's the observation we have. We like to study the microbiome in the stool for people who have coffee, don't have coffee, to see whether there are any differences. There is something out there. We know it helps for many other liver diseases. So that's maybe a window for us to think about how the diet and what we do may help diseases for which we don't have an active therapy. And so more along these lines we'll see coming out in the years to come. That's my expectation, but we can talk about this in the next session.
All right, I have some more rapid-fire nutrition if you're ready. So, Detox Tea or liver cleanses. Someone actually sent their Detox Tea up for you to see.
I am not going to recommend Detox Tea. This is a big thing being sold on the internet at this time. We actually had we've had a couple people doing it, and unfortunately I saw them in the hospital. I can't say it had to do with that. I would not purchase anything that says it detoxes or cleanses the liver.
They can have it back.
We'll dispose of that for you. So, high fructose corn syrup.
So as an overall part of a healthy diet, if you are a patient who is in the normal weight range or the obese weight range, then I would try to avoid high fructose corn syrup. I don't know that it plays any role specifically in PSC, but as part of a healthy diet. However, if you're in the underweight group, I would not hesitate to use whatever you need to use to maintain muscle mass and body weight.
What's better, low carb, Mediterranean, paleo, keto?
Okay, so I
does it
avoided
matter?
that topic, you guys, because it could be an entire talk in and of itself. The best general diets to follow, especially in your scenario, would be something along the lines of the Mediterranean or the anti-inflammatory diet. There are many problems with diets like the keto diet. I didn't hear what else you said, but where they're promoting either excluding an entire macronutrient from our diet or reducing a macronutrient part of our diet, and that would not be healthy. The liver needs all of its nutrition for various metabolic pathways that occur in cells, and excluding or reducing carbohydrates, such as in the keto diet, would not be a helpful thing for you all.
Okay, I'm still getting through all the fungal questions here. How about pharmacogenomic testing? Is there a role for that for PSC patients?
At the present time, I don't see there is a reason to, unless you take some other medications for which you worried about. But for the space where we're talking about PSC, we don't see any particular reason.
This one says, injections of chemicals into the gallbladder can cause PSC. Why don't we study the gallbladder? I think the gallbladder was just a delivery mechanism, just an anatomical way to get things to the biliary tree. Is that right? Can biospecimens be sent to Mayo from patients living elsewhere?
We can send you the kit at home. You can do it at your own convenience at your local lab, and we have done almost 80% of our collections for either Mayo patients or non-Mayo patients through this mechanism, so have great experience to do that. We'll be happy.
Is there a larger number of PSC in those who emigrate from Northern Europe?
There are no studies looking at the immigration pattern from Northern Europe, but there clearly is a huge gradient of PSC in Europe. So it's the #1 indication for transplant in Scandinavia. And by the time you get down to Italy and Spain, it's a very rare disease. And I've never really had that explained to me. There's a similar gradient, and Costas would know, there's a similar gradient in MS from bilatitude. That's been attributed to vitamin D. I don't think vitamin D has been implicated in PSC that we understand. I clearly have seen immigrants from Northern Europe in this country with PSC, but whether it changes coming to North America or not, there hasn't been a more recent immigrant wave to study that.
We have a large database with those 1,500 plus patients across the U.S., and we know where they lived for their entire lives. And we try to understand the South versus Northern question and migration within the U.S., and we couldn't find any patterns, #1. #2, we know as far as the work that John Eaton did when he was working with us, we were published these 1,000 questionnaires about diet. We found that Western diet associates more with PSC. And so high in fat, red meat and the likes, and people who eat more seafood, they're protected. What's mind-boggling to me is like the Norwegians, they eat a lot of fish and they have more prevalence. So there is more to it. There is more to it.
But maybe fish causes PSC.
Maybe.
Have any animal models been tried using germ-free mice? And what about stool transplant for PSC patients?
Yeah, hot topics. So first of all, Dr. LaRusso did a study where he found that germ-free mice have actually worse PSC-like disease in the mouse models. And so it's simply not that we have a microbiome, we have a healthy microbiome, it's there for a reason. Taking it away is not good for the liver. I think the data are clear. FMT, I think the data I've seen in IBD and PSC is marginal, and we cannot make any firm conclusions. I don't think the signal's been very positive, been very impressive either.
We need to understand the signature of the microbiome in the stool and other species in PSC patients and controls to better understand how we can manipulate those systems to have some therapeutic endpoints. I think one of the major advantages we have here, as Dr. Gores said, we have 3 major laboratories working in different aspects of the disease. And so as we make discoveries in one space, we pass them to the other group and we think globally, and that's a tremendous opportunity as we interact among ourselves to think broadly. And so those are very important questions you bring forward for us also to think forward where we go, what we should do, and we learn from you every time we interact in many different ways.
last question, I think. These patients are very generous. They want to donate their entire liver upon their death or transplant to you. Is that possible?
Quite frankly, we don't have an IRB written this way, but you know, we're open to see whether we have any thoughts. We have a lot of explanted livers over the years that we know we're using in many different ways. And so, as we said earlier, the organ of damage is the liver, and we don't have access to this, and we cannot do biopsies. So we have to think broadly about what we can do in a situation like this.
Okay. So we got to a lot of questions. There's a lot of dietary questions still here. I think Nickie will be around if you want to ask her directly.
So just one final comment for Nickie. Clearly, she's a Midwestern girl. The celery juice wasn't in her repertoire. But I'm so shocked you knew the cost of CBD oil.
It was hearsay.
Hearsay.
Yeah.
We're done.
Thank you very much, Doctors Huebert, Lazaridis, and Gores, and Nickie, Francisco Ziller. You gave us lots of food for thought, and that's not meant as a pun. It was a fantastic combination of talks, and we all learned so much. And I really appreciate that you put it down to a level where we can understand while still teaching us so much. It was fantastic. Thank you so much. Next, we have our traditional make-your-own yogurt sundae snack, which is sponsored by the Belmont family today. You have 30 minutes to enjoy your snack, visit our vendors on this level, and visit our PSC shop. And also, please go, if you haven't had your blood drawn, the phlebotomists are still in Riverview E today until 5 o'clock. The teens and college-age attendees who are going on the outing, you're going to leave at 10:15. I think that is it. Go have a good snack.